my slides. If you look at those slides, some of those, not all the slides, I put a notes there. So you can, this is for your further reading, and I believe some of them will help you understand the slides better, because maybe during the lecture I just catch something not really briefly, and then you can go back to read what's going on and what I really mean during my lecture. So first, I just want to introduce myself, and my name is Jijin. I'm the associate professor in the Department of Biological Sciences in this dental school. And I'm not a dentist, so here I just want to introduce you some basic sciences. I believe that's important for your future as a good, even better dentist. And currently I'm running two NIH-funded research projects. One is on the, we call it cancer immunology and cancer immunotherapy, focus on head and neck cancer, how those cancer cells produce some molecules to modulate the immune response, therefore to convert our immune response to become like pro-tumor. And the other project is related to the HIV, and because the oral cancer, incidence of oral cancer in the HIV positive population is on the rise. So my work is just want to see why is that, if there is any mechanism associated with that, and if we can identify any drugs or therapeutic approach to solve the problem. So I have my email and phone number here. This is my office. Where's my office? Oh, never mind, it's not there. On the third floor. And just walking around, you can find me. And so if you have any questions regarding my lecture, let me know. You can email me, you can call me if I'm in the office, and or just like walking around, find my office, then we can start talk. Today I just want to talk about some immune molecules focusing on cytokines. So cytokines are the molecules that interact with, they can mediate communication between different types of immune system and different types of immune cells. So we all know cell communication is important, just like neurotransmitters in the transmission of those electronic signals from one part of the body to the other part of the body, or just like hormones, they can produce different types of hormones, stimulate expression of different molecules involved in development and reaction to the environment. So those are two reading materials. I'm not really sure if you guys still using the Vital Source or just buy the books. So those are two books. I think that can be useful. But be honest, for the purpose of exam, and then you just focus my lectures. So those are the learning objectives. Today I just want to talk about properties of cytokines, not very details about what they are and their structures, not about this, and very basic properties of cytokines. And also I will talk about cytokine signaling, because cytokine signaling is important as we can identify so many different types of drugs or medicine in treatment of different immune diseases. And you will hear that cytokine signaling all the time in this body as a host. And finally, I'll talk about some cytokines. I believe those are important in the different types, like innate immunity and adaptive immunity. And also, if I have time, I'll mention about the regulation of the immune system, how the immune components are interact with each other in the body. So the first thing I just want to talk about properties of cytokines. And when we mention about the cytokines, the first thing we want to talk about will be immune cells, because cytokines are basically produced by different types of immune cells. And this is, I'm not really sure if Dr. Levin already mentioned about this. This is the rod of a stem cell. Becomes different types of progenitor cells in the bone marrow. Eventually, it will become different types of mature immune cells, including like lymphocytes through lymphopoiesis, and also different types of monocytes become macrophages or dendritic cells. And those are the cells, they produce different types of cytokines. Doesn't mean other cells, other immune cells also produce cytokines. However, those are the major cell types. They produce different types of cytokines involved in our immune system. So let's just put it in more details. For some reason, there was a little, I don't know why, but I didn't mean this means something. It means nothing, just like the other one. So we have lymphocytes, B cells, and T cells. In T cells, we have type 1 T cells and type 2 helper T cells. You know that, right? I just assume you know that, okay. So we call it Th1 cells or Th2 cells, and also we have natural killer T cells, NK cells. We have Th17 cells and a couple other cells. So you can see here, T cells are accounted for somewhere 14 to almost 50% of total white blood cells in our circulation. And those monocytes, they take over they only account for like 2 to 12% of white blood cells. Those are the cells and their function, they can produce different types of cytokines for the further development and execution of the immune response. Just like I said, all the other cells, like B cells, they can produce cytokines, and even basophil cells, they can also produce cytokines. I mean, even the main function of those cells, like for example, B cells, are produce antibodies. For those cells, they are their main function is phagocytosis. So I mentioned about hormones and neurotransmitters. What are the differences here? So those hormones can be small molecules, like a melatonin. Is this a protein, a peptide, or small molecule? Prostaglandins, so they are not proteins. And hormones can also be peptides, like insulin, leptin, those kind of hormones. For neurotransmitters, majority of them are small molecules, like different types of amino acids. Dopamine, they can be some small peptides, like somatostatin, oxytocin, and so on. However, all cytokines are proteins or peptides. Those cytokines are produced by cells involved in both innate and adaptive immunity. I already mentioned about. These are small secreted lung antibody proteins. They are not antibodies. They can mediate and regulate immunity, inflammation, and hematopoiesis. So I think like many years ago, now if you read some papers, those cytokines, they have different types of names, like a lymphokine, because they produced by lymphocytes to interact with other types of lymphocytes, or monokines. Those are the small peptide made by mononuclear phagocytes, and chemokines, interleukin, those different types of names, even like a tumor necrosis factor alpha, TNF alpha. So one thing, right now we just call all these different types of names as cytokines. They are all in the cytokine category. And if you, however, in literature and in your reading in those lectures, you will hear a chemokine and interleukin all the time because there's so many cytokines, they already bear the name and they will have the name forever. So you will hear TNF alpha. This is a very specific family of cytokines. And so you just need to know all these are in the cytokine category, so nothing else. So the other cytokines, they call it interferons, like type 1 interferon, interferon alpha, and interferon beta as a type 2 interferon. Interleukins will be IL-1, IL-2, IL-6, those kind of, those are all the cytokines. Chemokines including, there are two types of chemokines. One is called CXCL. This is a cysteine. In the middle of the cysteine, there are two cysteines. In the middle, can be any amino acid. This is called a cysteine X, cysteine like a ligand, or CCL cytokines, chemokine. It's a cysteine like a ligand. Those are chemokines. TNF alpha already mentioned about it, and hematopoietins will be EPO and CSF. So I just use the abbreviations just for easy for you to understand it. You really don't need to know that in details, but you have to know those listed here, they are called in the category of cytokines. So cytokines are produced in response to immune stimuli, and they are not stored preformed. So they are unlike some other proteins. They already have the protein inside the cells and stored in certain vesicles. Once the cell, you stimulate the cells, then they will release the protein. However, cytokines are different. So they produce in response to immune stimuli, and therefore, when the immune cells receive like immune stimulation, they have to start from the very beginning. From DNA get activated to produce mRNA, and from mRNA through the translation process to produce protein and secreted to the extracellular space. And in most of the time, those cytokines has to be modified further by either enzymes to get a glycosylation or from bigger molecules chopped to become smaller molecules. And therefore, as a result of that, they have relatively slow cellular response. Most of the time, you need about like somewhere 8 to 16 hours to see the cytokine increase. They can act on the cells that produce them. So this is called autocrine action. They can act on nearby cells. It's called a paracrine action. And also they can act on distant cells. And this is called a endocrine action. So this is cartoon clearly lists all the types of interaction of certain cytokines with cells. So you really need to know this is important because in the immune modulation you will see this kind of thing all the time. But we have to know endocrine action is really, really rare. So, however, those two, paracrine action and also autocrine action, those two are most common action for cytokines. And those cytokines, they can pleiotropic, so that means a cytokine, one cytokine can induce different biological effects. And they can be synergy. They have combined effect of 2 or more cytokines to produce even higher activation of the targeting immune cells. And they can be antagonism So one cytokine inhibits action of the other cytokine. Typical example will be IL-10. So IL-10 is anti-inflammatory cytokine. So if you treat cells or the targeting cells receive the IL-10, that will inhibit production of different types of cytokines. And the cytokines can induce other production of other cytokines. This is forms a cascade. So I'll give you one example about it. And the different cytokines can have similar actions. Like IL-1, TNF-alpha, they can also activate a similar signaling pathway to give you the same, almost the same cell response. So this is something really important. And this is our body to keep our immune system running. And then even you have one lack of one cytokine, but you can still have the other cytokine to do the job. And this is the cartoon to just summarize what I mentioned about different function models of those cytokines, like pleiotrophy, redundancy, synergy, and antagonism, and cascade. This is about the cascade, so you can look at it. It's very easy to understand. Also, this is important just because this is how our immune system is modulated by different types of cytokines. So I mentioned about redundancy, and that's important. So one example is here. If you look at this is a cytokine receptor family, and all those are like receptors response to IL-2, IL-15, IL-7, 9, and 4. So there is a common denominator for those. There is a common receptor for these receptor families members, and they all, even they are different receptors, they respond to different cytokines. However, they have IL-2 subunit, receptor subunit in the receptor. So if you look at IL-4 receptor, it has IL-4 receptor, but it has IL-2 subreceptor for signaling. So therefore, this receptor recognizes IL-4 as a ligand. However, the signal transduction and immune response will be very similar to IL-2, just because IL-2 receptor is there. And so defects, so actually you can see here, defects in a unique cytokine has a little effect on the whole immune response, immune system. Because, for example, if the body doesn't produce IL-7, you can still have IL-15 or IL-9 to have the same function. And this is a typical redundancy. However, defects in the shared component, or this is a common receptor, example here will be IL-2 subunit, and then this will have profound effects. This can be detrimental. So the IL-2 receptor gene is in the X chromosome. So defects in the X chromosome of this gene is called a severe combined immunodeficiency. And if you look at these mice, so this mice doesn't have IL-2 receptor, doesn't have this IL-2 receptor subunit. Therefore, those mice don't have T cells. They don't have mature T cells, and they are deficient in B cells. They don't have any immune response to those cells in these mice. So if those mice get infected by whatever pathogen, they will die immediately just because lack of the immune response. In human, this disease is related to the bubble boy. I think if you use Wikipedia or whatever, you can see there is a boy was born with defects in IL-2 receptor gamma subunit, and so therefore he was lived in like a tent has to be separated from environment. And eventually, he died just because I think his sister touched him or something like that, and then he died just because the immune deficiency. So, in the science field, these mice are very important for cancer studies. Just like in my lab, we can inject human cells into those seed mice. Just because they don't have an immune system, they will not reject human cells, and then those human cells can grow in those mice, this type of mice. So that's therefore these mice are very useful model for the cancer research. And only problem is, what else in the mice? In these mice, they are mouse cells and they are not human cells. So the other way we are doing that is we are generating humanized mice. So we will take out all the immune cells from those SC mice and put in human immune cells. Then those human immune cells can be repopulated in those SC mice that will mimic the human immune system in animals. So this is a really, really interesting study. That's what's going on in my lab. And I mentioned about those cytokines, they are proteins, therefore they have very short half-life. Right? Because the protein will degrade it in the circulation. And also they have very low plasma concentration, just because they can interact with specific receptors on the cell membrane and with really high affinity. So therefore, even you have very small amount of specific cytokine, then it can stimulate activation or deactivation of the certain immune cells. So when we are talking about cytokines in the body, we are talking about a nanogram per mil or per in the body instead of like milligrams or this kind of high level. Those are the sample questions I may put in the exam. Cytokines are antigen specific. Is that right or wrong? Cytokines are capable of activating more than one cell type. That's right, that's what we're talking about. And cytokines are made by lymphocytes and myeloid cells. Cytokines are small protein molecules. Cytokines are synthesized de novo in response to antigen or other cytokines. Cytokines are antibodies. So those are something I mean, my exam style, I will not ask you all the details because it's just simply not necessary. But you really need because I see people, the reason I believe those are very simple questions, to be honest. I couldn't make it even simpler, easier. So the reason I'm talking about this is just because I have some even residents working in my lab doing research, sometimes they get confused, cytokines or antibodies or antigens. And so I feel like I need to spend time to explain them just like I've been talking about to these people. So therefore, if you have very clear criteria, cytokines, there are cytokines, they are small molecules, you stimulate the cells, immune cells, they will produce it from DNA to the protein. This is a longer response time. Yeah.
Did you say C was true or false? Made by lymphocytes?
C is made by lymphocytes, like T cells and myeloid cells. That's true.
Oh, true.
Yeah. Those just because those are immune cells. So actually, non-immune cells can also produce cytokines. For example, a typical example would be like a T cell, cancer cells. So one of the reasons cancer cells are able to escape like immune attack just because they can produce some cytokines as growth factors for themselves, and also they have produced some like suppressive or anti-inflammatory cytokines to inhibit immune response in the body. So that will be a different story. These molecules are not cytokines. The A is cytokine, right? Complement molecules C3b and C5b. They are not cytokines, right? I never mentioned that. What about histamine? Because it induce whatever allergy. So is that cytokine? No, it's not a cytokine. Just because it's a small molecule and produced by mast cells. And TNF-alpha is a cytokine. It's a serotonin cytokine. Dopamine cytokine. This is a neurotransmitter. So we are clear, right? Any questions asked? So then I just want to spend some time to talk about cytokine signaling. So cytokine signaling, I just want to, first thing I want to talk about receptors for different types of cytokines. I mentioned about cytokine interact with respective cytokine receptors on the cell membrane with high affinity. So we have to talk about receptors. And I'll talk about more or less like a detailed signaling of NF-kappa B. How many of you know NF-kappa B? So you will know that. And also I briefly talk about STAT, JAK signaling pathway, and also chemokine signaling pathway. So cytokine signaling, think about it, it's a relatively, it's a very simple, right? So you have extracellular stimuli, like a bacterial infection or a viral infection or whatever injury that will stimulate those immune cells. Therefore, those induce cytokine gene activation, and then those cytokine gene, upon activation, those cells will produce cytokines in the circulation, in the environment. And then those cytokines will interact with cytokine receptors on the immune cell surface. Those are called the target cells to produce different types of biological effects like proliferation, differentiation, or even cell death. So the first thing I just want to talk about some receptors for different types of cytokines. And those are the receptors. Most of the time, we group those receptors by their structures. And those are immunoglobulin superfamily of receptors, and including IL-1 receptor or TLR, toll-like receptor family members. So I'm kind of feel difficulties here. So anyway, do you know what the TLR is? Toll-like receptors, T-O-L-L-L-I-K-E, toll-like receptor. Okay, so if you don't know much about it, those are the receptors on our cell membrane, and they can sense, say like a bacterial components, like bacterial cell walls, those kind of things, and stimulate cells to produce cytokines. Okay. Those are called the toll-like receptors. And IL-1, interleukin-1 receptor, we know that. And those two receptors, they are totally different receptors. However, they share very much similar structure. They all have, this is cell membrane, the extracellular domain, they have immunoglobulin structure. And in the toll-like receptor case, we have leucine-rich repeats. So outside of this, those structure difference, makes the wide IL-1 receptor is only recognized by IL-1. And the toll-like receptor is only recognized by bacterial components like LPS. If you look at intracellular domain, they are pretty much the same. So they all have toll-like toll IL-1 receptor domain, or TIR. So therefore, they will stimulate a very similar immune response because they will stimulate very similar signaling pathway to induce the expression of the same group, same genes. This is a TNF-alpha receptor family, and you can see those receptors form trimers. One, this receptor activated by TNF-alpha, and then it will recruit adapter molecules inside the cells to the receptor to stimulate TNF-alpha specific response. So you don't have to memorize class 1 receptor family, all these kind of things. You really don't need to do that, but I just want you to appreciate we have different types of receptors for different types of cytokines. And those are the receptors specific for GM-CSF, IL-5, IL-3, and these are the IL-2 family receptors. So they can recognize IL-2, 15, 7, and IL-9 and 4. I already mentioned about it, and they all share the IL-2 beta subunit. And those are the receptors, they all share a receptor subunit is called a GP130. So those are glycolytic protein, glycoprotein with molecular weight 130 kilodalton. So, so they have like a, so you can think about it because all those receptors they share GP130 subunit, they will trigger this very similar cell response in response to IL-6 and IL-11. And I mentioned about interferon, type 1 interferons, like interferon alpha and interferon beta, as well as type 2 interferons, interferon gamma. They trigger different cell immune response. And they have, therefore, they have totally different receptors. So for the type 1 interferon receptor, you have trimers, 3 different receptor subunits come together. Okay. And similarly, in the interferon gamma family, we also have 3 receptor subunits come together in response to the interferon. So one interferon interacts with the receptor, for example, here with interferon receptor 1, that will stimulate recruitment of interferon receptor 2B and interferon receptor 2C to form a receptor complex to stimulate signaling. And so again, cytokine receptors, so those are the chemokine receptor family. It's unlike previous other receptors I mentioned about. Chemokine receptor family members, they all have 7 transmembrane domain. So you can see they have 7 transmembrane domains. They have this kind of structure, and the binding of the ligand to the receptor just like this. So this receptor peptides in this receptor form this kind of barrier. So the signaling molecule, like for example, CCR2 will interact with the receptor inside of the receptor to stimulate the activation of the receptor. The other receptor family will be TGF beta receptors, and they are different from others. So I just list all the those major receptors for cytokine signaling, and you can read it, but I'm not going to put anything like that in the exam. However, want you to know these receptors are required for cytokine signaling. IL-2 receptor is right. What about this one? T-cell receptors. So they are not cytokine receptors, right? CD28 is CD28 a cytokine receptor. You know CD28, right? Because no. Yeah. So I thought so Dr. Nguyen might have mentioned about it. So CD28 is on the surface of the T cells, and it's involved in T cell activation. So actually, it's a receptor part of the T cell receptor. And what about CXCR4?
Yes.
This is what about toll-like receptors? So actually, toll-like receptors, they are not cytokine receptors because they can interact with like a human virus, not specific to cytokines. So it's specifically to LPS. You know what LPS is? Maybe I skipped too much. So this is a like a bacterial site. Yeah.
So toll-like receptors are not part of them?
TLR3 is not cytokine receptor, so I just want to mention. So here I just want to say IL-2 is, and CXCR4 is cytokine receptor. Others, they are not cytokine receptors. So the other question would be like this: T cell activation by T cell derived IL-2 can induce IL-2 production in the cells. So this is an example of endocrine effect. ABCDE, which one is that? A, right? So we all know apoptosis is totally different story. So A is the only right answer to this question. So now it's a little bit tough part of my lecture. I just want to give you a detailed signaling pathway about NF-kappa B. The reason for that is because NF-kappa B signaling is really important, and I have to say this is pretty much the most important signaling pathway related to this cytokine signaling and active function. Because when I think someday Dr. Aaron Weinberg will talk about innate immunity, and all the innate immune system, function of the innate immune system is depend on NF-kappa B signaling. Dr. Levin will talk about like activation and inactivation of different immune cells. So again, the NF-kappa B signaling pathway plays a role in those lectures they are going to talk about. So, and also I'll mention about other signaling pathways like a JAK-STAT signaling pathway and chemokine signaling pathway, as well as crosstalk between different signaling pathways, because we were talking about synergistic effect, why they are synergistic. But I just want to mention about there is a crosstalk between those different types of signaling pathways. So when we are talking about NF-kappa B signaling, the first question we need to know is what are NF-kappa Bs? So NF-kappa B are proteins, right? And NF-kappa B themselves, they are not cytokines. So they are transcription factors. You know what the transcription factors are, right? Because they can interact with DNA to stimulate gene expression. And in resting cells, those NF-kappa B molecules are sequestered by inhibitory called I-kappa B protein. And then they stay in cytosol in resting cells. Therefore, they are inactive. And there are many NF-kappa B signaling inducers, and including cytokines like IL-1 and TNF-alpha, or microbial cell components like bacterial cell walls, lipopolysaccharides like LPS, viruses, they can activate NF-kappa B, like retrovirus, and double-stranded RNA from different viruses. And like other virus will be HIV. And so if specifically, I can mention about that. And receptors, they are involved in NF-kappa B activation will be IL-1 receptors. And toll-like receptors, also called the TLRs, is involved in the innate immunity, and also TNF-alpha receptors. All of these receptors will stimulate NF-kappa B activation. So just like I mentioned about in resting cells, NF-kappa B is sequestered. Yeah.
Okay, so what does NF-kB tell the cell to do?
So I'll mention about it.
Okay.
And so it's involved in many different functions. Say like cell protection, apoptosis, and also stimulate production of other cytokines. Like specifically, it's involved in inflammation. So binding of IL-1 to IL-1 receptor stimulate recruitment of different types of cellular proteins to the receptor, intracellular domain of this receptor. The reason I list all those proteins here, just because those proteins are potential drug targets. They are potential drug targets. They really like those big pharma companies are working on those proteins to identify a specific way to block interaction of, for example, MyD88 to the other proteins, therefore to inhibit NF-kappa B activation. So most of the time, we really want to inhibit NF-kappa B activation just because NF-kappa B activation is involved in many different types of autoimmune diseases. So you want to inhibit it instead of activate it. So this is called a complex 1. And then once the complex 1 forms, so the receptor will disappear. So you only left the complex on the cell membrane. And then further recruitment of other proteins induce the phosphorylation of a key kinase called IRAC. This is a protein kinase, and it so induce the degradation of this IRAC, and then it forms complex 2. Then those complex 2 will interact with more proteins. So from the cell membrane, migrate into cytosol, where they get phosphorylated and recruit by other key kinases. One key kinase here is called a TAC1, TGF-beta activation kinase 1. It gets phosphorylated in complex 3. And this is a key event for activation of whole NF-kappa B pathway. So then phosphorylated TAC1 can interact with other accessory proteins, I-kappa B or NAMO. So eventually will induce phosphorylation of I-kappa B here. So those, those are like inhibitory. So this is I-kappa B kinase. And then I-kappa B kinases will phosphorylate I-kappa B in I-kappa B, NF-kappa B complex. Therefore, to induce I-kappa B, this is the inhibitory factor degradation. So since I-kappa B is gone, then NF-kappa B get activated. And then it will migrate into the nucleus, binds to the specific DNA, and activate expression of those genes like TNF-alpha, IL-6, IL-8, like a human beta defenses. And this is the only reason I mention about this, just because I believe Dr. Aaron Weinberg will talk about that. The epithelial cells in oral cavity can produce those antimicrobial peptides, and those induction of those antimicrobial peptides is related to the NF-kappa B, because if you think about it, and if you have any LPS or any bacterial insult in oral cavity, so the innate immune system, in other words, our epithelial cells will respond to it to activate NF-kappa B and to activate human beta defenses, so to protect our epithelial cells from those insults. And here I mentioned, give you the detailed map about NF-kappa B. I will not test this in the exams, but I just really want you to appreciate the complexity of a specific signaling pathway, and all those molecules involved in the signaling pathway can potentially be the drug targets. So the other signaling pathways are called JAK signaling, JAK state signaling pathway. So JAK is called the genus kinase. This is not a kinase. So this is not a cytokine. This is a kinase inside the protein kinase inside the cells, and also STAT, signal transducer and activator of transcription. So this is by the name. This is a transcription factor inside the cells, and those are the cytokines can induce activation of JAK STAT signal pathway, interferon alpha, beta, and interferon gamma, EPO, GM-CSF, IL-6, all these can stimulate JAK STAT signal pathway. So this is the reason that's important. I mentioned about NF-kappa B is activated by IL-1, by TNF-alpha. However, the JAK STAT signal pathway is activated by IL-6. In other words, IL-6 is not going to activate NF-kappa B directly, right? So there is a possibility IL-6 will activate NF-kappa B indirectly by stimulate the production of other cytokines. So this is a brief description of JAK state signaling. Again, in the resting cells, STAT is inactivated. So it just stays in the cells, but they are not activated just because they are not phosphorylated. So only phosphorylation of the STAT, this protein, will activate it. And once those cell receptor receive the signal from, for example, IL-6, it will activate phosphorylation of JAK. So the JAK phosphorylation will induce phosphorylation of STAT. Those are STATs are transcription factors. Once phosphorylated, so this phosphorylated protein will enter the nucleus of the cells and stimulate the transcription of specific genes like cyclin D1 to D3. Those are the cell cycle modulators, stimulate the cell proliferation. Chemokine signaling, there are two chemokine, major chemokine families, like I think I mentioned about it, CCL and CXCL. For the CCL family, we have CCL1 all the way to CCL 27, CXCL1 to CXCL14. And those are receptors totally different from the cytokine receptors I mentioned about. Those are T protein coupled receptors. They all have the cell 7 transmembrane domain. So in the resting cells, those receptors are inhibited by a subunit inside the cells called an inhibitory G-coupled protein. So therefore, it's the alpha receptor, alpha domain of this protein is inhibitory, so therefore the receptor is inactivated. The binding of the ligand, I use the CCL12 SDF as an example, will induce a dissociation of those subunits from the receptor. So therefore, the receptor gets activated to induce downstream signal pathway, including phosphorylation of PI3 kinase and MET kinase and PLC, stimulate target gene expression. Those genes are related to adhesion, polarization, chemotaxis. In other words, that will stimulate cell migration. So, I didn't mention about here. So some of the cells, like monocytes in the circulation, and eventually they will become macrophages. And however, you only see macrophages in tissue. How do those monocytes get there? So they can migrate just because you have, for example, you have SDF molecules produced by tissue, injured tissue. And then those monocytes will sense that, will go there and will migrate into the tissue. So this cytokine receptor is important, is critical for chemotaxis because the migration of those immune cells. And those are the also true example questions. Cytokines are nuclear transcription factors that induce gene expression. Yes or no? False or true?
Steve? I think that's true.
The question, and then I'll ask, do you think like cytokines are transcription factors? They are not, right?
They induce transcription.
So that's false. NF-kappa B is a transcription factor that can be activated by IL-1 and TNF-alpha signaling. This is true, right? I think that's true. In the absence of receptor activation, NF-kappa B is sequestered by inhibitory I-kappa B protein inside the cells. True or not? This is true. STAT signal transducers and activators of transcription proteins are always active even in the absence of interference. False. That's good. We're doing good. Do you want to take a break or just want to? I don't have a lot to talk about.
I guess it depends on how much more you.
I'll see how many slides I have.
Still quite a few.
Okay, maybe we can take a break. 5 minutes or 10 minutes? So I just want to continue the lecture, follow the lecture by cytokines and the regulation of the immune system. And again, I'll focus on cytokines because just like I mentioned about the numerous times, cytokines are important modulator in our immune system. So basically, I mean, definitely we'll talk about like antigen, recognition of antigen, and presentation of antigen by antigen presenting cells, and also whenever that happens, you will have cytokine involvement. So here, I just want to this is a very simple way to categorize those, classify different types of cytokines. I just just want to try to make it very simple. But it's not. It doesn't mean you will, when you read papers or something else, you will have some other things about cytokine category of different types of cytokines. So here I just want to mention about mediators of innate immunity, mediators of adaptive immunity. And in the cytokines, I mentioned about pro-inflammatory cytokines and anti-inflammatory cytokines. They are, so it doesn't mean there is no clear cut between, so just like the others. So one thing I want to mention here is there is no clear cut between mediators of innate immunity and mediators of adaptive immunity. So some of those cytokines, mainly they act in the innate immune system. They can also interact with immune cells in the adaptive immune system. And also I mentioned about stimulators of hematopoiesis, because those are the cytokines that are involved in white blood cell differentiation and proliferation. So NF-kappa B, TNF-alpha. And so TNF-alpha is produced by activated macrophages and T cells. TNF-alpha is very important pro-inflammatory cytokine. And so it's produced by LPS membrane bound toll-like receptors. And so actually TNF-alpha is also can be induced by IL-1. And TNF-alpha mediates recruitment of neutrophils and macrophages to site of inflammation. Therefore, this is a pro-inflammatory cytokine. It can stimulate endothelial cells and macrophages to produce chemokines. So that means then those cells can attract or induce migration of circulating immune cells to where you have the injury or insult by bacterial proteins. And also, this is a very potent pyrogen causing fever by direct activation or through IL-1. So this is whenever we have a fever, then we can see we have some TNF-alpha in the body at a higher level. And it can stimulate production of acute phase proteins, such as like a CRP. And it has roles in rheumatoid arthritis, psoriasis, and many other diseases. So in this case, just because we have too much TNF-alpha. So the drugs on the most of the drugs on the market is inhibitors, like a monoclonal antibody against the TNF-alpha. And this is a cartoon, just simply summarize the role of TNF-alpha in our immune system. So it's involved in macrophage activation, lymphocyte development, hematopoiesis, and also thrombosis in our body. So this is a very important cytokine. IL-1 is produced by activated macrophages, stimulated lymphocytes, keratinocytes, like epithelial cells, and fibroblasts. It can activate the NF-kappa B signaling pathway. So I talked about NF-kappa B signaling I used IL-1 as the cytokine to activate the signaling pathway. So it has this very similar effect like TNF-alpha. It helps activate the T cells. It can be induced by inflammation, injury, and infection. So that's very important cytokine. IL-2 is involved in adaptive immunity. So IL-2 is produced by activated T cells most of the time. B cells, NK cells, monocytes, macrophages, and a subset of resting T cells, they can also produce IL-2. IL-2 signals through the IL-2 receptor. IL-2 has key function of the immune system, tolerance and immunity. This is a growth factor. It will stimulate the T cell proliferation. So once T cells get activated by interaction with antigen-presenting cells, those T cells will produce IL-2. And then those IL-2 will activate IL-2 receptor on the same T cells to produce even more IL-2. So this is a typical autocrine effect regarding IL-2, and it stimulates T cell proliferation. And also it promotes T cell differentiation. So those T cells will we call the naive T cells become effector T cells and memory T cells, like helper T cells. There's a cytokine drug, it's called a Proleukin on the market to treat renal cell carcinoma and melanoma. This is a synthetic IL-2. Currently, it's on the market for this cancer treatment. IL-12 is also a mediator of adaptive immunity. So it's produced by macrophages, dendritic cells, T cells, and NK cells. So when we are talking about the cytokines, we need to know what cells produce them and what kind of families they are in, what kind of signaling pathways they can stimulate, and then what kind of biological function they will have. So this is important because they can stimulate production of interferon gamma. And interferon gamma production in T cells, specifically in helper T cells, is a hallmark for Th1 differentiation. So you already know like what the Th1 cells are, right? Do you know what the Th1 cells are? Helper, type 1 helper T cells. So those are the T cells they produce interferon gamma. And also they can enhance differentiation of cytotoxic T lymphocytes, CTL. So SS1 in periodontal disease, IL-12 may mediate alveolar bone loss. So this is some study have been done in, I believe, somebody's lab in our school. IL-10 also is a mediator of adaptive immunity, but this is so I mentioned about TNF-alpha, IL-1, they are pro-inflammatory cytokines. And IL-10, on the other hand, is anti-inflammatory cytokine. Okay. So primarily, this is an inhibitory cytokine, and it's cytokine produced by Th2 cells. That's the type 2 helper T cells, and or produced by activated macrophages. It inhibits interferon gamma production. So this is a totally different from IL-2. IL-2, let me see, totally different from IL-12. IL-12 stimulates interferon gamma production. but IL-10 inhibit interferon gamma production. Therefore, it will switch type 1 interferon become type 1 helper T cells become type 2 helper T cells. Okay. And that's really important. And it can inhibit production of many different type cytokines in macrophages. IL-4 is produced by macrophages, type 2 helper T cells, and activated B cells. It has a really complicated biological action, and so it enhances antigen presentation of B cells to T cells, because B cells are considered as antigen presenting cells to activate B cells. And also, it will stimulate differentiation of Th2 type 2 helper T cells from naive helper T cells. And this is important. It can stimulate Ig class switch from IgG1 to IgE. And IgE is related to allergy. And also is present in chronic and aggressive periodontal diseases. It's destructive to the tissue. Type 1 interferons are interferon alpha and interferon beta. They produce primarily by macrophages, also produced by many other cell types. Yeah.
Okay, so interleukin 4 induces the creation of more Th2, which then induces what?
So I don't want to say, well, on the surface, yes. And so it will induce it because it will inhibit interferon gamma production. So those Th1 cells no longer produce interferon gamma. And then since they don't produce interferon gamma, eventually they will become type 2 helper T cells or Th2 cells. That's one thing they do. The other thing they do, IL-4 is doing, is to stimulate Ig class switch. So cells from IgG1 expressing cells to become IgE expression. Those are the antibodies. IgG1 and IgGE, those are antibodies. And so the IgE production is associated with allergy.
So, but periodontal disease is not an allergic reaction, or is it?
No. But this is an immune disease, like it's called an autoimmune disease.
Oh, okay, got it.
So we have like an activation, uncontrolled activation of the immunity in the local environment.
Okay.
So that's why the cells get destroyed, not by bugs, but by cells. And also interferon alpha and beta can activate NK cells. Type 2 interferon is produced by Th1 cells I mentioned about, and it can activate NK cells and increase. So this is, I just put it here, and I believe in Dr. Levin's lectures, he will talk about this increase in MHC1 and MHC class 2 expression to have Th cells and antigen presenting cell interaction. Okay, so this is something he will talk about. And this is important to promote B cell differentiation to plasma cells, because we all know B cells produce antibodies, right? However, once those B cells can produce a lot of antibodies, those are the B cells, we call them plasma cells. So interferon gamma is important in promotion of B cell differentiation to become, those cells will become like antigen producing plasma cells. And also will promote cytotoxic T cell differentiation. And I mentioned about those cytokine, interferon gamma involvement in the periodontal disease. So, those are the based on the research, not clinical studies, just for your reference. Transforming growth factor beta. This is a cytokine produced by T cells, macrophages, and other cell types. So other cell types, including cancer cells. There are 30 members in the family, and they have effect on many different cell types. Basically, they can inhibit proliferation of T cells and activation of B cells. This is inhibitory. So I understand sometimes we get confused about type 1 helper T cells and type 2 helper T cells. I thought Dr. Levin mentioned about it. However, I think this is just if you already know, this is a summary of what we need to know. If you don't know that, this is a very brief description to tell you what are type 1 helper T cells, what are type 2 helper T cells. So Th1 cells, they are T cells, they can produce interferon gamma, IL-2, and TNF-alpha. So they are involved in cell-mediated immunity and phagocyte-dependent inflammation. However, Th2 cells, they produce IL-4, 5, 6, 9, IL-10, and IL-13. IL-10 is anti-inflammatory cytokine. Yeah.
No, I thought the IL-4 is what induced the Th2, but the Th2 is what makes the IL-4? So it's like a loop?
This is a loop. Yeah.
Okay.
They consume the cells produce IL-4.
Okay.
And so that's here. That's interesting because it will. I want to say there is no specific sequential action. We have IL-4; those cells will become Th2 cells. You really need many other factors together to give you the effect. So in this case, you also need IL-10 because IL-10 inhibits interferon gamma production, and only Th1 cells produce interferon gamma. So without IL-10, even you have IL-4, it's not going to switch Th1 cells to Th2 cells. I'm not really sure. I think that the slides I uploaded doesn't
That's
have
what we
this.
don't have.
Right. So I put it here. Just want to help you understand what's the difference between Th1 and Th2, and also just because the reason we say those are Th2 cells or those are Th1 cells, most of the time based on their ability to produce what kind of cytokines they are producing. So, and also therefore the functions are different, and the Th2 cells are involved in inhibition of several functions of phagocytic cells, and so therefore induce phagocytic independent inflammation. I just wanna summarize my lecture. So I mentioned about properties of cytokines and what are they, and how do they interact, and how do they act, stimulate signaling pathways. So that's why I mentioned about the different types of receptors, and also mentioned about the NF-kappa B signaling pathway in very detail. And so I mentioned several cytokines, and they are important for T cell differentiation, macrophage activation, and they are involved in innate immunity and adaptive immunity. So those are the cytokines. I mean, those cytokines, they have very a variety of clinical applications. I mentioned about here as for your reference. Okay, one example will be EPO. We know that like some athletes, they are using the EPO drug to enhance their performance. Interferon gamma is involved in treatment of multiple sclerosis, interferon beta. So IL-2 for kidney cancer and melanoma. TNF-alpha monoclonal antibody has been used for treatment of IBD and rheumatoid arthritis. Two drugs important. Famous will be like Humira and infliximab. Those are monoclonal antibodies to reduce TNF-alpha levels in patients. So any questions? If you have any questions regarding the lecture or specific questions about whatever I didn't make it clear in the lecture, just email me. So you already have my email there. And any questions? Any questions?
Could you go back to the slide on the differentiation between the TH1 and TH2 cell?
Okay. Is there any way I can forward this just this specific slide to you?
Or you can take a picture of it.
Yes, you
I
can
mean,
send
I
it to
just
the
was
team
missing like
drive.
3 words. So
Okay.
Who's gonna take it to the-
I believe this is also will help you understand what